This animation depicts a dark, frozen landscape of abstract cellular structures. In modern biology, we can sequence a cell's DNA, and we can take incredibly detailed static snapshots of its interior using advanced electron microscopes. We cannot, however, film the live molecular processes of an entire cell continuously in 4 dimensions. To get around the physical limits of microscopy, researchers took a radically different approach. They built a living cell entirely inside a computer. Their subject is JCVI-SYN3A. JCVI-SYN3A is a synthetic bacterium engineered to contain only the absolute minimum number of genes required for life, possessing exactly 493 genes. To understand how this organism functions, the researchers constructed a 4D whole cell model. A 4D whole cell model is a unified digital framework that calculates how an organism lives, grows, and divides across 3 spatial dimensions and time. Understanding life at this level requires mapping the biology directly into a software engineering architecture. It requires reverse engineering the math of a living organism. The foundation of this simulation is a mathematical chemical engine. Before the cell even has a physical shape in the computer, ordinary differential equations calculate the raw synthesis of lipids and the building blocks of DNA. Ordinary differential equations are mathematical formulas used to compute continuous bulk rates of change over time. But a cell isn't a perfectly mixed test tube. For a chemical reaction to occur, molecules must physically bump into each other in 3-dimensional space. This reveals the spatial layer. A 3D lattice strictly limits where densely packed gray proteins and orange RNA move. Neighboring panels show the enormous DNA volume projecting onto this grid, creating severe bottlenecks that dictate how quickly ribosomes assemble. Survival in this environment depends entirely on the successful spatial navigation of molecules through a highly restrictive grid. The final layer of the computational stack governs the physical dynamics of the chromosome itself. This cross-section models DNA as elastic green and magenta polymer chains. The computer calculates physical forces to prevent strands from illegally crossing during train track replication, which causes massive entanglement. Below, a precise 12 piconewton force partitions these chromosomes into distinct daughter cells. The true test of the model is running all of these layers simultaneously. The chemistry, spatial diffusion, and polymer physics operate in tandem to progress the cell through a complete 105-minute biological life cycle. Generating just a few of these simulated cells requires hundreds of hours of computation on GPUs. GPUs are graphics processing units, specialized circuits capable of handling thousands of parallel calculations at once. Synchronizing these distinct mathematical solvers produces the first continuous 4D map of a cell transforming from a single entity into 2. To test the boundaries of this digital organism, researchers simulated 50 replica cells. They gave every single run the exact same genetic code and identical starting conditions. No 2 cells behaved identically. At the end of the 105-minute cycle, every single run resulted in a completely unique cellular composition upon division. The variance comes from stochasticity. Stochasticity is the inherent chaotic randomness of physical interactions that occur at the microscopic scale. This split-screen visualization tracks how microscopic chaos scales up. Random spatial encounters on the 3D grid cause tiny delays or accelerations in chemical reactions, which compound drastically over the life cycle. A genetic code is highly specific, but it acts only as a recipe. The actual realization of that code relies on the unpredictable physical dance of molecules colliding in space. While this model only covers 493 genes, it provides a stable platform. It gives researchers a foundation to start layering in the more complex reaction networks found in naturally occurring, more sophisticated organisms. By digitizing this minimal cell, we can see how the progression of a biological state is inextricably tied to the underlying physics and chemistry of its environment. It shows that even in its simplest form, life is a product of how its individual components navigate a crowded 3-dimensional world. If you want to explore more about how computation is decoding life, like and subscribe to Computational Genome Biology.